Hello friends, welcome back to our YouTube channel Doctorscorner. Today we are going to discuss about tubuloglomerular feedback mechanism. Okay, it is basically a mechanism which will regulate the GFR, that is the glomerular filtration rate. So what exactly is this mechanism is, in a nutshell, any changes in the renal tubular sodium chloride reabsorption, okay, that is changes in the renal tubular sodium chloride reabsorption, okay, sodium chloride reabsorption will causes actually change in GFR, will causes change in GFR, that is any increase or decrease in the renal tubular sodium chloride reabsorption will causes increase or decrease in the GFR. Okay, that is the basic crux of this tubuloglomerular feedback mechanism. To understand this concept in detail, let us go to some basics. Okay, I am just drawing a nephron. Okay, a nephron, a tubular structure of the nephron, a line diagram. So this is what I am drawing is the Bowman's capsule, which will continuous as proximal convoluted tubule. Then we have a loop of Henle. This is loop of Henle. So this loop of Henle is continuous above as distal convoluted tubule. Okay, so for this I will write DCT for distal convoluted tubule. Then finally it is continuous as collecting duct. Okay, now the important structure here is this blood vessels here, which forms the glomerular capillaries. Okay, glomerulus. So this glomerulus, basically it has afferent arteriole, that is from where the blood is coming, it is known as afferent arteriole, from where the blood is coming, that is afferent arteriole. Okay, so let me draw this a bigger one. So this is afferent arteriole. So it is continuation from the various branches of the renal artery. So ultimately it will subdivide into so many structures, and finally afferent arteriole, through which the blood will come, and it will passes through these glomerular capillaries, and it will pass through efferent arteriole. Okay, so it will pass through efferent arteriole. So this is efferent arteriole. I hope you know this basic anatomy of the nephron. Okay, now basically the filtration that takes place in this nephron is the glomerular filtration, right? Now you can see the filtered fluid will go through this tubular structure, proximal convoluted tubule, loop of Henle, distal convoluted tubule. In this distal convoluted tubule, that is early part of distal convoluted tubule, the epithelial cells of this distal convoluted tubule, which I am drawing with the blue color right now, it is specialized near the afferent and efferent arteriole. It has a specialized epithelial cells. That particular patch or plaque of this epithelial cells, it is termed as macula densa. Okay, macula densa, very important for our today's discussion. Macula densa, what is this? Basically in the wall of the distal convoluted tubule there are specialized epithelial cells. Okay, similarly I am going to show in the green color, there are specialized cells here in the arteriolar wall, that is in the afferent as well as efferent wall near this macula densa, it is near the distal convoluted tubule. These specialized cells are known as juxtaglomerular cells. Okay, so together they form the juxtaglomerular apparatus. So we are not going to discuss about that concept today. We are now sticking to tubuloglomerular feedback mechanism, but to understand this we should know about this macula densa as well as juxtaglomerular cells. Okay, now there are two mechanisms here. So basically about this tubuloglomerular feedback mechanism. So the two mechanisms are afferent arteriolar mechanism and efferent arterial mechanism. But before that, just have a quick look to the diagram here. So you can see this is the distal convoluted tubule, and this green color structures is this specialized epithelial cells of the termed as macula densa. Similarly, in the afferent arteriole and the efferent arteriole, which are in close contact with this distal tubule, the smooth muscle cells here in the arteriolar wall are specialized, and these specialized cells are termed as juxtaglomerular cells. Okay, that's what I have described you. Now this is the correct diagram for this. Now So as I said, this one, it has two mechanisms. It is tubuloglomerular feedback mechanism. One is afferent arteriolar feedback mechanism. Afferent arteriolar feedback mechanism. Another one is efferent arteriolar feedback mechanism. Efferent arteriolar feedback mechanism. Okay. So what is this efferent arteriolar feedback mechanism is? Coming back to this diagram again, as I have already highlighted you about the macula densa. So basically it will sense this sodium chloride level, okay. So in this tubular level, sodium and chloride level it will sense, okay. So any increase or decrease of the sodium chloride level which is sensed by macula densa can alter the lumen of afferent and efferent arteriole. Either it can increase or decrease the afferent arteriole level or efferent arteriole level, okay. So how it is possible, let us discuss now. So see, I will just draw a flow chart to understand this thing. Suppose there is decrease in the blood pressure, that is decrease in the arterial pressure, that is the blood pressure. So whenever there is a decrease in the arterial blood pressure, there is decrease in the glomerular hydrostatic pressure. Hope you agree with this point, okay. So glomerular hydrostatic pressure, glomerular hydrostatic pressure is decreased. So this will ultimately lead to decrease in the GFR. Okay, hope this is very clear to you. That is, decrease in the blood pressure ultimately causes decrease in the glomerular filtration rate. Now, glomerular filtration rate means the filtration at this level, that is this one, GFR, what I am writing, filtration at the level of this glomerular membrane. So normally it is 125 ml per minute. So decrease in the blood pressure causes decrease in the GFR. So now, if there is a decrease in the GFR, then what will happen is the macula densa, which is present in the distal tubule. So the sodium chloride level in the macula densa is decreased. How exactly, it is not known, but certain experimental studies suggest that decrease in the GFR slows the flow rate. Okay, suppose there is a decrease in the GFR. So this decrease in the GFR, what it will do? Decrease in the GFR, it will slow the flow rate. It will slow the flow rate. Okay, it will slow the flow rate of the fluid in this tubule, and because of this decrease in this flow rate, it will cause us increased reabsorption of the sodium and chloride, which is delivered to the ascending limb of the loop of Henle and ultimately to the DCT. So when there is a decrease in the sodium chloride level, which is sensed by the macula densa, so it will send signal to the afferent arteriole. Okay, it will send signal to the afferent arteriole. So how it will send signal to the afferent arteriole, and what it will cause is, so ultimately there will be dilatation or increase in the diameter of the afferent arteriole. So afferent arteriolar dilatation is seen. Dilatation means, here I will write resistance is decreased. So when the afferent arteriolar resistance is decreased, so the diameter will increase. So ultimately what will happen is, whenever there is a decrease in the sodium chloride level at the macula densa, so it will sense, and what happens is there is increase in the diameter. Okay, increase in the diameter. So suppose initially the diameter is this much, so after that the diameter will increase. So resistance is decreased means the diameter will increase. If the diameter of this is increased, afferent arteriole, then naturally the blood flow will increase, and ultimately the decreased GFR is controlled, that is there is increase in the glomerular filtration rate. So this is how sodium concentration in the macula densa will control the glomerulus. That's why it is called as tubuloglomerular feedback, that is tubules are controlling the glomerular filtration rate. Okay, any change in the tubule is causing the glomerular filtration rate. This is the afferent arteriolar feedback mechanism. Now there is efferent arteriolar feedback mechanism also. What is that? I will just draw this in the flow chart. So see, efferent arterial mechanism is similar here. So you know there is juxtaglomerular cells present in the efferent arteriole. I have already highlighted. It will release renin in response to decrease in the sodium chloride level in the maculadensa. This renin, by the renin-angiotensin mechanism, it will convert angiotensinogen into angiotensin 1 and ultimately into the angiotensin 2. Okay, this angiotensin 2 will causes the constriction of the efferent arteriole, that is efferent arteriolar resistance is increased. Efferent arteriolar resistance is increased. So through this mechanism efferent arteriolar resistance is increased. So by both this mechanism, what will happen is, if there is increase in the diameter of the efferent arteriole, it will causes inhibition of this decreased glomerular filtration rate and ultimately GFR is increased. Similarly, if there is increase in the efferent arterial resistance, it will, what it will do, it will causes the inhibition of this decreased GFR. So that is, it will causes increase in the GFR, okay, that is decrease in the glomerular hydrostatic pressure. Okay, so just let me revise what we have discussed today. We have discussed about the tubuloglomerular feedback mechanism. What is this tubuloglomerular feedback mechanism? Is any change in the sodium chloride level in the renal tubules. Renal tubules means here in distal convoluted tubule will causes changes in the GFR. How? By two mechanisms, efferent and efferent arteriolar mechanisms, feedback mechanism. So whenever there is a decrease in blood pressure and decrease in GFR, maculadensa will sense decrease in the sodium chloride, which will cause as dilatation of the efferent arteriole and constriction of the efferent arteriole and increases the GFR. Hope you liked our video. So please subscribe our channel, Doctor's Corner.